So here is prophase of mitosis, in which the nucleus, which is not shown here, but it dissolved, and the DNA inside the cell, this is the cell, condensed into these chromosomes. And one chromosome is consisted of two sister chromatids, and you see these two are homologous pairs, so they're identical, and so are these. So there are two homologous pairs in the beginning, and that is prophase. In metaphase, now we have our homologous pairs, we have two of them, and they're going to go into this sort of linear formation here. So they're going to line up in the middle of the cell like this, and we actually have two centrioles, which are on either side of the cell, and these spindle fibers will begin to form out of microtubules, and they'll begin to form and sort of connect to each chromosome. So just to sort of show you metaphase again, these are the spindle fibers that form coming out of the centrioles and attaching to the chromosomes in this sort of linear formation. So now here we have anaphase. So the spindle fibers have attached to all the chromosomes, and now the sister chromatids in each chromosome separate, and they get pulled apart by the spindle fibers like this. And after the chromatids separate, like they just did in anaphase, then they're going to separate into two different nuclei which are going to form, and then the entire cell, the rest of the cell is going to split, which is called cytokinesis. So now you're left with two daughter cells, and the chromatids sort of unwind back into their non-condensed form. So you end up with two daughter cells, and you can see there are 4 separate strands of DNA in each, so that's 2 diploid daughter cells, and that's mitosis. Okay, so now we're in meiosis, and so this is prophase 1 of meiosis, and you'll see it looks very similar to prophase of mitosis. So we have our 2 pairs of homologous chromosomes, like these two, that's one pair, and these two are another pair, and the nucleus has dissolved. But in prophase 1 of meiosis, it's a little different than prophase of mitosis because something different happens. Synapsis occurs, so these two homologous pairs are going to combine, they're going to form this structure called a tetrad, and something called crossing over occurs, in which the genes from this maternal chromosome are going to mix with the genes from this paternal chromosome, and I'll show you what they look like after that happens. So now we have crossing over, it's occurred. So you see, there's bits of, red inside what used to be just blue, and bits of blue and what used to be inside just red of the chromosomes of what I drew, which really means that some of the genes from this chromosome, from the maternal chromosome, have mixed in with the genes from the paternal chromosome. They've swapped out. So now we have these new unique non-identical genes. So that's prophase 1 of meiosis. So now in metaphase 1 of meiosis, in mitosis and metaphase, the chromosomes lined up in this sort of linear pattern that happens here in meiosis during metaphase 1. The only thing is they stay in their tetrad sort of structures. They don't separate to make a line of 4 chromosomes. They stay in these structures. And then those spindle fibers are going to form from these two centrioles. So now we're in anaphase 1 of meiosis, and in this stage, the spindle fibers have attached to the chromosomes, and they're going to separate these chromosomes from their tetrad structures like this. Okay, so now we are in prophase 2 of meiosis. So that big cell that the genetic information was just in, the nucleus split, and so did the rest of the cell. So now we're left with 2 cells. So that was the first cell division of meiosis. There's 2 of them. So that was the first cell division. Now we have 2 cells. The nucleus, which had been formed, has redissolved in prophase 2, and that's what happens in prophase 2. And then we're going to go on to metaphase 2 of meiosis, in which again, the chromosomes are going to line up like so. And you don't see the centrioles on here. I'll put them in on the next shot, but you're going to have the centrioles on either side in each cell, and those spindle fibers, again, are going to start forming. Okay, so this here is anaphase 2 of meiosis. So you can see here we have our spindle fibers again, and we're separating these genetically unique sister chromatids apart again, and it's going to look like this. So here we have the end result of meiosis. This is from telophase 2, so those two cells have each split to form an end result of 4 daughter cells, and these 4 daughter cells are haploid, meaning they only contain 2 strands of DNA each, as opposed to the 4 strands of DNA contained in the daughter cells resulting from mitosis. And so that was meiosis and mitosis.